labeling. This is the hypoblast here, and this is the epiblast layer, and the amnion, which is the membrane of the amniotic cavity, and the hypoblast is throwing an membrane which is forming the secondary yolk sac here. And this is the thickening in the hypoblast, which is a future mouth, which is known as precaudal plate, and extra embryonic mesoderm. It's the splanchnic layer of extra embryonic mesoderm, and this is the somatic layer of extra embryonic mesoderm and chorionic cavity. Can you see the depth of this? This is the disc, and see in the next view what happens. And they're going to show you the disc. This is the amniotic cavity, and this is the yolk sac. And this is that bilaminar disc in the second week. You can see the rounded appearance of the disc. Embryo at this stage is in the form of this disc, having this blue layer epiblast and this yellow layer as the hypoblast. The epiblast and the hypoblast here, this is the oval disc which is going to form the embryo. They have removed the rest of the chorionic cavity, just showing you the amniotic cavity and the yolk sac so that you can see how the gastrulation process takes place. Now, this is the upper view of the embryonic disc, the embryo, right? And this is the, now you can just see the epiblast here, and the amniotic cavity is here, and the yolk sac downwards. A groove or a streak will appear in the epiblast. That streak is actually formed by the migration of epiblastic cells in the center. Just see that migration here and the formation of that groove. This is labeled as the cranial end, this is labeled as the caudal end. How it has been identified, we have identified it, the first thing which identified or labeled the cranial end was a precaudal plate. The precaudal plate defined this end as a cranial end because it's going to the future mouth. But when the streak appears in this region, this is the caudal end. That is a second point which helps to label the ends. The caudal end, you will see now, this is the appearance of a streak. On the caudal end, there is a streak or a groove which has been formed, and this end is the cloacal membrane, and this end is the precaudal plate where the oropharyngeal membrane will appear. Now, this is the embryo, which is a disc shaped in this with a primitive streak here. The cells from the two sides will migrate and dip in the streak, and these are the epiblastic cells which will grow downwards in the underneath the this primitive streak, underneath this epiblast. They'll show you this view. Oropharyngeal membrane, which is the precaudal plate, the primitive streak, and the cloak. They're now going to cut this to show you the two layers. Now see this section here. Now this, they have cut at the level of the primitive streak. And this end or terminated end of the streak is known as primitive node. And now you can see this blue is the epiblast and this yellow is the hypoblast. The cells are migrating from here into the streak between the epiblast and hypoblast. See that. Next, they're going to zoom this, and now they have shown you the two layers, the epiblast and the hypoblast. And this was the point of the primitive streak. I'll show you the dipping in of the cells. Those cells which will be migrating from epiblast, underneath this, it will replace this hypoblast into endoderm and will create another layer of cells in between these two layers, which is going to be the mesoderm. See here. The cells are moving from the primitive streak, and they have from the primitive streak, and they have first replaced the cells of the hypoblast, and this is going to form the endoderm, definitive endoderm. Next, Intraembryonic mesoderm is going to be formed. The same from the cells on the primitive streak, they are migrating, and now they'll create another layer between this epiblast and this endoderm. This is intraembryonic mesoderm. And now this epiblast is labeled as the ectoderm. This is labeled as the ectoderm. Right, I'll show you just this view again here. Now this is labeled as ectoderm, which was initially the epiblast. After the formation of the endoderm and intraembryonic mesoderm, this is now known as ectoderm. So three layers have been formed. This is ectoderm, intraembryonic mesoderm, and endoderm, and it's a trilaminar germ disc, and the process of gastrulation has taken place. This is one thing in the third week. There's so many things which happen in the third week, but the first step is the formation of is the trilaminar germ disc. The process is gastrulation. This is the primitive streak here. This end is the cloacal membrane, and this end is the oral pharyngeal membrane, and this is the primitive node here, right? And now this is the blue layer is known as ectoderm. Embryo is like this at this stage, right? As it gradually grows, it will fold and give a shape of the embryo. All the components of this diagram, the epiblast, that should be labeled as ectoderm now, the cranial end, the primitive node, the primitive streak. Now see what is happening here. एक स्टेप दोबारा वापस जाएं. They want you to see now that the intracells migrating underneath this region, right? And now this is going to form intraembryonic mesoderm under this ectoderm or epiblast. Initially, it is the epiblast. When it forms intraembryonic mesoderm, the leftover cells are the forming the ectodermal layer. The cells are migrating first here, then the cells will migrate cranially, but they leave a middle portion. And why do they leave a middle portion? Because a group of cells, this region is the primitive node. This node grows as an extension cranially underneath this to form a rod-like structure here, which is known as notochord, which is actually going to form the body axis. Right? They'll show you this. Now view this again. Now this is they have removed the epiblast, and they have shown you the second layer and the third layer. The endoderm is complete, but the mesoderm, which is intraembryonic mesoderm, is still in the process of formation. It has formed caudally. Now it will grow cranially and form a complete sheet, leaving a small portion in the midline where there is a central cord, which is the primitive axis, and that is known as notochord. See here. The cells from the primitive streak, they are migrating all around. They have just separated first here. Now they're migrating in the cranial region, and other process which is growing from the from the primitive node, that is growing in the form of a cord here, and the intraembryonic mesoderm is on its side, and it will grow like this, and this is the notochord. The notochord process grows and it is forming a notochord. Now, this is how intraembryonic mesoderm has been created. The middle layer is a red layer which has been created by the primitive streak, and its primitive node has created a notochordal process. And see what has happened to the primitive streak. After the formation of this intraembryonic mesoderm and replacement of the hypoblast as the endoderm, the primitive streak has decreased, it regresses, and it disappears. After formation of this trilaminar germ disc, it will disappear. That is the fate of primitive streak. After the formation of the endoderm, the mesoderm, and ectoderm, and the notochordal process, it will disappear. Then the next thing, now they have come back to the its surface view to show you another process, and that is formation of a neural system, the nervous system, the neural tube, and that is coming again from this ectoderm. This is the primitive streak, which is now decreasing and it is disappearing. This is the cloacal part, and this is the oropharyngeal membrane. Within the ectoderm, one region dips in to form another tube. It folds in. We will